Okay, so now in this third part of chapter 3, we're going to look at how the cell divides to make more of itself. Okay, so the life of a cell is called the cell cycle. The cell cycle, yes, just think the life of the cell. Now the cell cycle, only this part here is mitosis. This is when the cell is trying to divide. But the rest of the cell's life is called interphase. So the interphase contains, which they labeled here, G1 when the cell is growing after it has divided and become a new cell. The S part of interphase, this is when it's going to make 2 copies of DNA. Okay, so again, interphase, G2, here it's the normal growing. But then after G2 is done, then the cell can get ready for the cell's life when the cell is not dividing. Okay, so here the cell grows, does its normal functions, but so importantly, here this is when the cell replicates its DNA or makes 2 copies of DNA to prepare for mitosis. It's making 2 copies so that the 2 new cells that form after it divides can each get a copy of DNA. Okay, so in interphase, the different parts during that, there's the G1, G2, those are for growth. The S part of interphase, just being more specifically, this is when DNA is replicated, so 2 copies of DNA are made. Okay, so interphase is when the copies of DNA are made, so the mitosis is when we get to divide those 2 copies of DNA. And then cytokinesis is when the cytoplasm just splits off, or the cell splits off into 2. So I'll just write that down here. Cell splitting into 2. The different steps of mitosis. Prophase is the first part. Know that during prophase, the DNA ends up turning into these shapes, these X shapes. Okay, so what these X shapes really refer to is each line of the X is 1 copy of DNA for this 1 chromosome. And we have a total of 46 chromosomes. Okay, so each line, if you think 1 copy of the chromosome DNA, so then it adds another 1, and so now you have the 2 copies. Okay, so prophase is when these X shapes are appearing. Metaphase is when we're going to see that the chromosomes line up right down the middle of the cell, those X shapes. Then anaphase is when those 2 lines of the X shapes separate from each other. So the chromosome separates and then move in opposite directions. And then telophase just kind of looks like when the 2 new cells are trying to appear. Okay, so know the major things that happen during prophase, metaphase, anaphase, and telophase, and be able to recognize them from pictures. So interphase, here the DNA just looks convoluted. It's just in these long strands. But in prophase, so now the DNA is arranged into these X shapes. Here the chromosomes are now visible. And we're also seeing here the centrioles. Centrioles, if you remember, are those yellow rods for cell division. And the centrioles are giving off microtubules. These lines, this is called this mitotic spindle, but really just made up of microtubules, so think of them as ropes. Prometaphase is in between prophase and metaphase. Okay, but don't worry too much about prometaphase, but then in metaphase, the X shapes are right in the middle. In anaphase, the 2 lines of the X's have separated, and now they're moving to the opposite sides. And the mitotic spindle, those ropes, are pulling them in the opposite direction. And if you look at telophase, it looks like the cell is trying to split into 2 after the DNA has separated from each other. And in cytokinesis, the cytoplasmic division. And so this really just refers to, more specifically, the cell membrane trying to split up. And so this really starts in anaphase. Okay, but really just know the definition of cytokinesis. Cell division is regulated by our cells. So the cell is only supposed to divide at certain times, only certain cells are supposed to divide. So for example, our skin cells divide a lot, intestinal cells, blood-forming cells, but Neurons, just an example, neurons only divide when we're young, when we're babies, kids. And then neurons stop dividing, or that's what we know so far anyway. Maybe there might be a new scientific discovery about that, but neurons don't divide. So you can see how it's not all cells that are supposed to divide. And so hormones, growth factors are regulating cell division. Normal cells are supposed to stop dividing if they become too crowded. Okay, so if there are already cells around them, they don't want to make more. Okay, so that's called contact inhibition. But unfortunately, cancer cells do not care. Cancer cells want to keep on dividing. So cancer cells are abnormal. They should stop dividing. They should make more of themselves, but they don't care. They want to make more of themselves. So let's talk a little bit more about the tumors. There are two types of tumors. Benign tumors remain in the local area. Malignant tumors, malignant cancer cells, they travel to other organs. And they can use blood vessels or lymph vessels to do that. Oncogene. Know that an oncogene is an abnormal mutated gene in the DNA if it increases mitosis and causes the cell to become abnormal. Tumor suppressor genes. So normally these are the ones that are supposed to look to see if the cell is normal and they would stop mitosis. But if they themselves become mutated, then they're no longer controlling anything. And so then this can lead to cancer cell as well. But I believe on the study guide and on the test, I would ask the oncogene. Know that an oncogene, abnormal form of gene, that leads to a cancer cell.